Okay. Today, we have a session on ecotoxicology and pollution biology by Mr. Vasantha Kumaran, and he is a research scholar in National Taiwan Ocean University, Keelung, Taiwan. And he have a tremendous knowledge in the fields of zoology, and he completed his master's in zoology from Tamil Nadu. And currently, he is pursuing his PhD in marine biology, and his work is basically on the ecotoxicology in the marine species like crabs as well as the corals, and he have published more than 15 papers related to ecotoxicology and pollution biology. A warm welcome to Mr. Vasantha Kumaran for our session, and we are inviting you at Eco Foundations for a talk. Thank you, Vasant. Please, you can start the session now. Yeah. Hello, Vasant.
Am I audible? Hello.
Yes, sir, you are audible.
One minute. Okay, am I audible now?
Yeah, yes, sir, you are audible.
Yeah. Shall we start?
Yes.
Hello, Vasant. Hello.
Hi.
Yep.
Sir, yeah, I'm waiting. I'm
Actually, there
unable
are
to share this.
screen. Okay, let me share this.
1 minute.
Let me allow you to share. Can you try it now, Vasant? Now your camera is on.
But I couldn't share my screen. Can you see my screen now?
Yes, sir, I can see.
Can you see now?
Yes.
Yes, sir, entire screen is visible
Okay,
completely.
sir. Okay, sir. Okay, just a sir. Yes.
Yeah, Vasant, the problem is there is a lot of noise.
Around me?
Yeah, from your side, there is a lot of noise. I don't know what is it. Are you typing?
Some typing, yeah,
Yeah,
typing sound is there.
Now it's okay?
yeah. Now it is fine.
Yeah,
Maybe can you make sure that it is in the power slideshow mode?
it's okay now? Yeah.
Yes. Yeah.
Good evening to everybody. So, as our director and founder mentioned, I'm going to take a lecture on ecotoxicology and pollution biology. So, my talk will be more preliminary and very in old school style, so you can able to follow all the information I give. It will be more like a class. So, since the talk is about ecotoxicology and pollution biology, so we can know first what is toxicology. Toxicology is a branch of study which concerned with the nature, effects, and detection of poisons. See, the toxicology can be study about any toxins or poisonous material, even a external foreign body also. It can be study about the vibration. It can be study about of excess air, excess noise, excess chemical exposure, pesticides, heavy metals, and so on. So it can also, the study of toxicology gives us a great measurement and analysis of toxin and intoxicating banned substances and prescription medication present in person's body. So these are the branches of toxicology, like environmental toxicology, aquatic toxicology, forensic toxicology, clinical toxicology, and regulatory toxicology. So, environmental toxicology is just the study of chemical effects on ecosystem, pollution monitoring control, mainly on plants, animals, and other microbes. Coming to aquatic toxicology, effects of pollutants on aquatic organism, and it also shows the water quality parameters like water quality assessment, like how much pH, how much temperature, how much chlorophyll content is present and changed due to the toxins which is exposure in the water. Coming to the forensic toxicology, it help us to detect the poison and drugs in body fluids, and it also helps in knowing the criminal investigations and legal evidence for toxic exposure. Clinical toxicology is a diagnosis and treatment of poisoning, therapeutic drugs monitoring, patient care in hospital and emergency settings. So there are more toxicological terms, so it is more or less connected to everything. So I will go one by one, so you can understand of that. Okay. As environmental toxicology, like industrial waste, agricultural chemicals, airborne pollutants, there are lots of other foreign external matters which affects the environment. So not only the bigger animals or higher animals get affected by toxins, every small and microbes are getting affected due to the man-made toxicology things. So generally the toxins or toxicants are done by either animals or human beings. So coming to the aquatic toxicology, there are many test organisms like fish, brain shrimp, crustaceans, and algal for knowing the accumulation of toxins or toxicants in a particular organisms or a particular water system. So with fish toxicology, we can know the behavioral studies with the fish. We can know the enzymatic activity and survival studies in the crustaceans. In algal, we can know the growth and pigmentation studies and lipid analysis, and we can come to a conclusion how much the organism get affected by the toxic exposure. There are many kind of pollutant types like heavy metal, pesticides, herbicide, industrial chemical, oil spills, and hydrocarbons. Like heavy metals, we can know for few example, there is lead, cadmium, even chromium. There are lots of heavy metals which cause toxicity to living beings. So cadmium is one of the most toxic heavy metal. So I will cover it later in our talk. So there is two kind of toxicity, acute and chronic. There is bioaccumulation studies, there's biomarker analysis, and ecological risk assessment. Um, Even aquatic toxicology gives knowledge on environmental monitoring, pollution management, and regulatory guidance. So these are some terms or terminology in toxicology, so which you should know as a basic to study about the um, any kind of toxicology, maybe animal toxicology, aquatic toxicology, even in low level cytotoxicology. You should know these kind of little terms to know to do the experiments or the science. Toxin. Toxin is generally it's a natural occurring toxic substance produced by living organism. For example, snake venom. It is a toxin. Uh, Bacterial toxin. It is a kind of a toxin. And plant alkalines. Everybody would have said that plant is a very good thing. It is a healthy thing. If you consume that, it is very good for our as a nutrition. It's very important to our nutrition. But it is not like that. Not every plant is healthy. The plant has the most poisonous substance. Some of the plants has the most poisonous substance like alkaloids. So it is very very crucial to examine before we consume that or before we are getting exposure to plants. Coming to toxicant, it is more or less like it is a synthetic poison which is otherwise called as man-made poison, uh, generally like pesticides, industrial chemicals, and other pollutants. Poison, any substance which is natural or synthetic that causes harm, it is called as poison, like cyanide and etc. Xenobiotic. Xenobiotic. Okay. Xenobiotic is nothing that like um, the syrup, the cough syrup, which we consume every day, the tablets which we consume, or any other prescriptionized medicines are called as xenobiotics. This actually, because of the manufacturing companies, it is leaven out in the aquatic system. The xenobiotic is a major pharmaceutical drugs, you can call like that also. It is a major threat to aquatic toxicology, aquatic lives, and it is transferring to even humans now. This is another part which is called as ADME concept, which is absorption, distribution, metabolism, and excretion. Like it is like a full on process how a toxins or toxicant enters into our body or excreted or distributed or metabolized in our body. Okay, so absorption, like as the term say, it is entry of the toxicant. The route of the entry is oral, inhalation, and dermal. Uh, Coming to the distribution, the movement of toxin through the blood, tissue, and other organs like liver, kidney, brain, or it is distributed through these organs. Uh, it is the primary part of the ADME. So it is distributed to various organs like liver, kidney, brain, and other even the visceral fat. Not only the normal fat we have, even the visceral fat. The visceral fat is that the fat which is covered by our organs. Okay, which is called visceral fat. Okay, even that fat gets exposed to the chemicals which is around the environment. As metabolism is biotransformation of these toxicant, there's like two phases, which is oxidation, reduction and hydrolysis, and phase 2 is conjugation. Like this oxidation and reduction plays a very important role in toxicology. Why? Because oxidation and reduction comes in the bioassay as oxidase and reductase. These two terms comes very in all the biochemical, biomarker, and antioxidant enzymes activities. So you will be getting more knowledge about these oxidase and reductase in the following slides. And as excretion is the eliminating the toxin from our body by urine, fecal, bile, sweat, and exhalation. It also affects our kidney function, our pH level, even our creatine level in our urine. So it is a major concept which is we are covering here. These are the absorption of the toxicants. Here you can see oral, by the skin, dermal. So as you all know, there are three type a layer of skin. So it also goes through the dermal thing and respiratory thing, like gases, dust, fumes, and other things. Injection, like venom, drugs, experimental chemicals, all those stuff. And musical absorption. Okay, like 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 our like in our nose, in our eyes, in oral mucosa also. This is how the excretion of the toxicants happens. So urinary excretion, fecal excretion, pulmonary excretion, gills. Generally we have lungs, right? So the aquatic organism have the gills. Through that, it excrete out. So generally, when we do the biochemical analysis, we will take gills and hepatopancreas for knowing the toxicology assessment. So it is the primary part, gills and hepatopancreas, because it accumulates most of the toxicant which is exposed in the water. Now we are going to see about dose response relationship. As you know, relationship between the dose and the toxicant, it shows the magnitude of the effect. Uh, the key parameters for that is the threshold dose. It is called a minimum dose cause effect. ED50, LD50. The ED50, LD50, LC50, or IV50 are the major toxicology terms which you should know before you are studying about the toxicology stuffs. So, as I mentioned before, LD50, which is called as lethal dose. The definition for that is the dose of substance causing death in 50% of the test subject. It is the unit of the LD50 is mg per kg body weight. The calculations done by various methods. I have given one example, which is called probit method. Then LC50, which is lethal concentration. Like lethal concentration is a substance in air or water causing death in 50% of test organs. For example, if you are doing a toxicology assay or toxicology experiment on a fish, on a crabs, even on a insects, you should, even on a mice or a rat, you should know the LC50 value. Okay, after knowing the LC50 value, you can fix the dosage for per group. Like if you're doing an experiment in lead or a cadmium, you will have a control group of that animal, and other two group, if like cadmium 50 percentage, like cadmium 0.5, cadmium 0.25, and cadmium 1.0. You should, you will be getting the information according to the LC50 value, so that it is you can know how much the toxicant is toxic to the animal. Then EC50, the concentration of causing a defined effect in the 50% of test organism. Then in toxicology, there is two type of studies. One is acute study, another is chronic study. In acute study, acute means short, acute means short, chronic means long. Okay, acute means like 24 to 96 hours, which means 4 days. Like we will assess the parameters every 24 hours till the fourth day. 24 hours, 48 hours, 72 hours, and then 96 hours. Usually, the acute toxicity, the dosage will be high. So, we can know the rapid onset and severe symptoms on the organism which we are testing. So, in the nature conditions, the too much poison like cyanide or any other poisoning material is will give us so much toxicity within short number of time. So for knowing the acute toxicity, as I mentioned before, we should know the correct LC50 and LD50 value, so that we can design the experiment according to that. So throughout the experiment, there should be no death in the animal groups. So we need LC50 value, so we can prolong the experiment till the end without any death in the animals. If there is too much of death in the animals, we cannot do the experiment and we cannot take the experimental organs for the other biochemical and biomarker studies. So we should know the sublethal dose to do in the acute study. For the chronic study also, it is more or like that only. But it can be, the word chronic itself says that long term. It can be prolonged for the week, months, for even years. Um, but the dosage will be generally low, and it will be repeated exposure. Like after 1 week, there will be more. We will give more exposure to the organism. After week 2, after week 3, every week you will update on the dosage. So there will be a optimum toxic conditions for our study to be done. So this is chronic and acute study. And in chronic and acute study, we can know the different type of stress that organism grows through. In acute study, if you see, there will be sudden shift and down regulation in the stress markers. In chronic studies, it will be reaching a threshold of the toxicity, and then it will be the organism will get adapted. So according to that, we can do different assays, not only assays and other experiment like microbiology assays, which is called microbiome. Then we can do transcriptomics, we can do genomics, we can do proteomics, like that. We can do different assays in chronic toxicity and acute toxicity. We can compare both. As I mentioned before, sublethal dose, the dose that does not cause death but may cause physiology and behavioral and biochemical changes. This is what I was mentioning in the previous slide. Okay, so we should know the sublethal dose, where if we are doing a experiment in a fish, we should know the sublethal dose, so fish should not die in between the experiment, so we can know the correct amount of experimental studies we are doing. Then we come to threshold dose, minimum dose at which the measurable toxic effect begins to appear. For example, lowest dose causing mild liver enzyme alteration in and like how to say, the threshold dose the name threshold. After threshold, it is the optimum. After threshold, there will be mortality in the animals. So knowing the threshold dose, we can see the like major complication in the organisms. So threshold dose help us to know that. And toxicokinetics and toxicodynamics. Okay, this is also major term in toxicology. So toxicokinetics, what the body does to the chemical, how our body reacts to the chemical, how our body getting exposed to the chemical, at which point, at the which threshold, at which sublethal dose, how our body reacts to the chemical, how it gets effect, or how to say, how to it bounce with our body or bounce with the organism. It is called toxicokinetics. It's like the key process of that is absorption and distribution. As I said, ADME, which I mentioned before, it shows the ADME, and ADME shows the toxicokinetics. It is correlated with each other. Then toxicodynamics, what the chemical does to our body. It is uh, vice versa to toxicokinetics. The study of interaction of toxicants with targeted sites like DNA, RNA, enzymes, receptors, different. So with this, we can know the oxidative stress, cellular damage, apoptosis, biochemical markers, much more. The factors affecting toxicity. Age. In younger age, there's higher sensitivity to immature organs. So in adults, there's compared to the young ones, in adults, it is standard sensitivity. In elder ones, there's a reduced detoxification and excretion. Like in younger, like in juvenile, in order to in fishes or insects, the juvenile will have disformed organs or immature organs, so that it will be more toxic to the younger ones. In the adults, in humans we are adults, in fishes we call it as brute stock. In that, the toxicity will be, the organism can withstand the toxicity of the toxicant, so it will be more or less, it has more capacity to bear the toxicant compared to the younger. In elderly one, which is more matured one in organism, there will be a reduced detoxification and excretion. So in humans we excrete every day, right? So excretion is a main form of detoxification in our body. We should excrete, detoxify every day to be healthy, so that when we get older, or when the organism get more older, there will be a reduced detoxification process and excretion process. Coming to the species, the factor affecting toxicity will have like different parameters, like absorption, distribution, and excretion. It may cause toxicity in every organism. It is not same. It for like in fishes, compared to the fish and a crustacean, fish get more affected by all the external factors, whereas in crustacean, crab get more having more power, or more withstanding ability against a toxicant. But in the shrimp, it has doesn't have the same capacity of the crab. Genetics. As genetics, everybody has different gene expression, gene expression, so that it is not same for every organism. The gene will be different. the gene expression will be different. So according to that, the stress will be affected to them. And other environmental factors like temperature, humidity, pH can also be one of the stress causing factors. Then bioaccumulation. It is it is a term which is very much preliminary and primary and very much needed in the toxicology subject. Bioaccumulation is accumulation of a chemical in an organism over time faster than it is eliminated. So you can see this picture so that you can get an idea on that, on bioaccumulation. The mechanism of the bioaccumulation is uptake via food, water, air, and it is stored in the fat, liver, or tissues. It is very, very slow metabolic activity, and excretion leads to the accumulation. Example: mercury in fishes, other heavy metal in the marine mammals, and DDT in birds. Like, we are coming to the birds, like there's a drug which is given to the cattles. It is directly affecting the vultures. Okay, if you know about that subject, you will know. So there's different mode of transmission of toxicant or poison to one organism to another organism, or one environment to another organism, or one organism to the another environment. So it is like, it is a full circle. Bioconcentration, uptake and accumulation of the chemical from water alone. It is observed across gills and skin of the organism, and the lipophilic chemicals stored in the tissues. Example, again, mercury in fishes, cadmium in crustacean, and PCBs in algae. Biomagnification. Biomagnification is like increase in chemical concentration at higher tropic levels in food chain. So if you are studying about a lake, if you are studying about a particular site area in an ocean, if you are studying about a particular site area in a river, or else you are studying about a particular mangrove system, you will know the biomagnification upon the species lives in that particular area. So if you are studying about a ocean sample site, you can take a organism from there, the crabs, the fishes, the algae, the zooplankton, the copepods, or other minute microbes. You can take them, and you can assess what kind of stress, what kind of other parameters the particular sample site area goes through. Like, in a sample area like a lake or in a ocean, if you take a crab, if you take a fish, we can do biochemical biomarker studies. We can know what and all heavy metals, pesticides, other xenobiotic like pharmaceutical drugs, which is in that area of our sample site. So with this organism, we can know the bioaccumulation things. So for example, there will be a very toxic area. There we cannot doing the fishing or proclaim the crab from that particular area. So we should not think that we should not take samples from there, because it is not directly related to aquaculture. Because we will take fishes or crabs for as a food, we will proclaim from very healthy, like very what to say, very like very amplified area for taking for proclaiming these species. So we should not think that we should take the crabs and fishes from that particular area, even it is very toxicant or it is not connected to the aquaculture. We should take some organism which is from the very polluted area, so that we can assess what and all the water parameters of that polluted area, and how much it is got toxified due to the outer substances. So biomagnification is very much renowned toxicology term. So it is not only in the aquatic thing, in other terrestrial areas also. So this is oxidative stress mechanism. So as I said, the oxidative stress as the oxidative stress is a very important terminology in toxicology. It shows us how much ROS production happen in a organism. ROS, like ROS is a reactive oxygen species. If for example, if we humans, okay, if we humans get stressed due to the other factor, even in our studies, in our personal life, if you are getting stressed due to the other food materials, if you are getting stressed due to the climate, ROS production will be happen. So ROS production happens in every organisms. So reactive oxygen species is a when there's a external body or foreign body exposure in our body, when it gives stress, ROS starts to produce. So there are so many kind of ROS, like superoxide anion, hydrogen peroxide, hydroxyl radicate, and singlet oxygen. The effects on the cell are repeat peroxidation. So there are different kind of um, stress level. Like in lipid peroxidation, we can know about the membrane damage. In protein oxidation, we can know how the enzyme inactivates. We can know about how much enzyme it is, the level of the enzymes in that particular organ or a species. We can in DNA damage study, like genotoxicity. There's assay called Comet assay. We can know how much DNA damage happened in that. This is another one is cellular apoptosis or necrosis. Like in very smaller organism like copepods or in uh, zebrafish embryo, we can do this apoptosis study like with the help of different kind of dye, which is AO dye, ethium bromine. There is a different kind of staining process which is involved in this apoptosis. So we can know the um, morphological defects happen in that particular species. This is next one is antioxidant defense. This SOD is the first level of defensive mechanism. This is CAT, which is catalase. This one is GPX, which is glutathione peroxidation. These are the different antioxidant defense systems which is present in most of the living beings. So you can see this flow chart. Then comes, as I mentioned before, SOD, CAT, and GST. Superoxide dimutase or dismutase is a first level of defense mechanism in a living being. The primary function is converting superoxide anion into hydrogen peroxide. Location of this one is cytosol, mitochondria, and extracellular. As I mentioned, the main importance of this SOD is it is the first line of defense mechanism against ROS, which is reactive oxygen species. Then catalase. The catalase is the second, we can mention it as a second level defense mechanism in an organism, which converts hydrogen peroxide into water and oxygen. So importance of the catalase is prevents H2O2 induced cellular damage. So if you are getting exposed to the very toxic substances, it prevents H2O H2O2 induced cellular damage. So with this, which we can apply in other animals, we can know how much H2O2 induced cellular damage has been happened. Coming to the transphase, it is actually the glutathione is a group which we can call it as, there are some different kind of glutathione antioxidant enzymes like GPX, GR, GST, and then GOT, GPT. So there are more number of enzymes which we can, which is very helpful to assessing the detoxifying effects of the toxicant exposure bodies. So G, with GST mainly we can know the pharmaceutical drugs toxicity in a particular animal. As I said, with biomarkers, these all are biomarkers. There are n number of biomarkers. These are the some preliminary biomarkers and very usually used biomarkers which we have, which I also working, which our lab also working here. It is a biomarker concept which is helpful to knowing the toxic effects or monitoring environmental monitoring in a particular area, or we can know priorly how the humans will get exposed to these chemicals. Okay, so that it is a biological indicator used to measure the exposure effect or sustainability to a toxicant. There are types of, these are the types of biomarkers, which is SOD, CAT, LPO, GST, GPT, GR, LDH, ACHE, and a lot more. So these biomarkers is very primary studies in toxicology. If you are taking aquatic toxicology as your study, you will do all these biomarker studies, which is SOD, CAT, LPO. It is a preliminary enzyme activity or biochemical activity to assess the toxic effects of a particular aquatic animal like fish, crab, and like other arthropods, all those stuff. So with this biomarkers, we can study about the biochemical, physiology, and pathology changes in a particular organism. It also indicates a sensitivity due to the genetic or health status. As I mentioned before, it is one of the major concepts in aquatic toxicology. As I mentioned, SOD cat before, this is ACHE, which is neurotoxicity assay, which shows how the neuro cells or neuro cells or neuro lining is affected by the external toxicant. Yeah. It- yeah. Like, yeah, ACH, again I will repeat it. Okay. ACH is, it is called as neurotoxicity, acetylcholine esterase. In our, even in our brain there is a nerve cleft. Okay, if the nerve have the nerve is affected by the extra foreign body, it creates a in a cleft, it creates a nerve synapse. So this acetylcholine esterase shows us how much the foreign body affects the brain or our spine or all those stuffs. Okay, so generally we will do this assay in crabs. In crabs we don't have definite brain. Okay, in crabs we don't have definite brain, so we will take eye stock for this ACHE assay, and we will take hepatopancreas and gills, because as the previous work shows, the hepatopancreas and gills have neuro cells or neural linings in the tissue, so we can assess ACHE in these things. In fish it is very easy to assess ACHE, because we can, in fish we can dissect brain. With the brain we can come to know the effect of ACHE. So this is the preliminary neurotoxicity. There is like BCCHE, there is CCCHE, many kind of neurotoxicity biochemical assays which you can do in your scientific career. As I mentioned, the test organism we can use fishes, crabs, copepods. We can use brain shrimp. We can use rotifer. We can do even in mice brain. We can do these assays. As I mentioned, lipid peroxidation, superoxide dimeters also a defensive biomarker. See, these are the model organisms: brain shrimp, copepod, zebrafish, chickembryo, rotifer, roundworm, mollusca. All these stuffs are toxicity testing models which can be used. And many people also do toxicology study in rat with rat also. And we can do toxicology study with nematodes. If you are a zoology student or an animal science student, you must have known about nematodes. Uh, there's a very highly important nematode called C. elegans. It also used as a very good model organism for toxicology. Then, uh, for the information, the person who did work on a C. elegans got the Nobel Prize last for the zoology. After that, nobody got Nobel Prize in zoology for in after he only got the Nobel Prize in zoology working on the nematodes, which is C. elegans. So, comparing the capacity of fighting against the toxicity of the toxicants, each species, each animals or different kind of animals, humans, even a dog, everybody has a different kind of defense mechanism. So, comparing fish, crab, and shrimp, fish have a moderate sensitivity to the heavy metal and pesticides. But in crab, it has more tolerance to the pollutants than the fish. So crab as a the outer shell, exoskeleton, which has a chitin and chitosan properties in the shell, it acts as a major defense mechanism because it can most of the pollutants or toxin cannot penetrate through the crab shell. So it is we can know that comparing to the fishes, crab has less more tolerance in the pollutants in the aquatic system. And in shrimp, as I mentioned before, the tolerance or the sensitivity can be depend upon their species and age. These are the some toxicology terms which I have mentioned before. If you want, you can take a screenshot. I will give you some more toxicology terms, like more like 30 or 40 toxicology terms which you can study and know about. So if you are going to a toxicology study or a cytotoxicology study in nanobiotechnology, in biotechnology, it will be more easier for you to know what and all parameters you can go through, what and the terms you can go through, what and all dosage you can set for your experiment. It will be very, very easy. So since I want to show very, very basic things, I have shown these things. We have more advanced studies and advanced presentation for this. But since this is our first talk to the introduction of toxicology, I am showing like just little terms. Um, this is this will be the I have taken some introductional part on toxicology and how toxicology works, what is the study of toxicology, all those stuff. Tomorrow we can see about what is pollution and the pollution impact on global level. So today we can finish it. Today we can finish it with this. So if you have any questions, you can ask me.
Hello, dear participants. If you have any questions, then you can ask to Mr. Vasant. Yeah, we are open for the discussion, and we have around 21 students today. So, if you have any questions, then you can either put that in the chat box, or you can just turn on your mic and speak. Okay. Vasant, there is one question for you. Does nuclear exposure comes under toxicology? And another one is also there. Do you usually do the studies on animals like zebrafish because their behavior are well known, and that any deviations can be easily identified? Vasant, maybe you can read. Maybe you can again read the questions in the chat box.
I will read it. Yeah, I will read it.
Okay.
Yeah, like as Melvin asked, zebrafish is a very good animal model because it is directly the zebrafish is. Can you share something about it? Yeah, zebrafish is a very good animal model for doing the toxicology study. It is a well-known toxicology animal model all around the world. So in my masters and even now we are doing a zebrafish toxicology study because it's more related. The If you study, you know that there are more number of things which is related to our human and other toxic or other aquatic organism. With zebrafish we can almost study every behavioral and every digestive physiology, stress physiology on other aquatic fishes also. So instead of studying about a particular fish, like in a freshwater ecosystem or in a marine water ecosystem, if you want to know the collective information of a particular toxicology effect, you can study on the zebrafish. So it will be more related. It will be more concisive to know how much a particle or a toxicant will can affect a aquatic organism. So zebrafish is a uh, good animal model. As Arundhati asked, can you share? Yeah. So I have like I have studied various toxicology studies. Like I have published a paper in Chemosphere, which is a developmental toxicology studies on zebrafish embryo, which is Daniel Radio. It is mainly studied on the cadmium oxide nanoparticle toxicity, how it affects the zebrafish embryo. So we took zebrafish embryo by spawning of female and male zebrafish. After getting the embryo, um, we should maintain the zebrafish embryo in the embryo buffer. So with that with that we have done some toxicology studies which I mentioned, no, the biochemical markers and biomarker studies like SOD, cat, LPO and GST, and major biochemical like protein and glucose. And we also saw the developmental stages on different days from 24 hours to 48 hours to 72 hours to 96 hours. We have seen the morphological defects in the embryo of the zebrafish. So, uh, like, uh, so this you can see if you search my name, or I will send the paper to the director. He can share you all about my previous study. Any other questions, sir? Amita has asked the difference between LC50 and LD50. Like I mentioned before, LD50, like LD50 stands for lethal dose. LD50 is the like amount of that particular material or given at once causes death of 50 of a group in a test animals. So LD50 is one way to measure the short-term poisoning potential of the acute toxicity of a group. Generally, the toxicologists use many kinds of animal, like I told you, zebrafish and other brain shrimp. Um, so with that LD50, we can know how much amount of material given at once for cause of death of the 50 percentage of the animal in the group. So generally, it is done in the milligrams. So when and she is asked about the difference from LC50 to LD50, no? So coming to the LC50, LC50 value usually refers to the concentration of the chemical in a water or a air, but in the environmental study, it can be mean the concentration of the chemical in the water, how much concentration it can be in the water or in a air. Like, wait a minute. I'm, I'm searching a slide for showing the LC50. I will display a graph which can be easily. Wait a second. In this is a graph of LC50, like LC50 in a marine crab. Okay, it shows the graph here. The optimum. This is a study on a temperature stress. Okay, it shows that the experiment has done in 25, 30, 35, and 40. Okay, these are the parameters which has been done before the experiment, so we can know the optimum dying conditions of the particular species. So in the 40th degree, in this LC51, in 40th degree there's 100% mortality rate in 4 hours. So the the experiment has been designed without this 40 degrees Celsius. So there should be less number of species dying in the groups. So we are, to say it simply, we should take off of this for doing our experiment, or we should know the sublethal of this thing after doing the calculation. So with that LC50 value, we can know the how much concentration, um, like we should do for the experiment. Any other questions?
Hello, dear Vasant. I think there is no further questions from the students, and I don't see anything in the chat box. But if the students have any question, then they can turn on their mic and they then they can ask, or you can ready for the questions tomorrow. You can bring your questions tomorrow. He will be available tomorrow as well for the second sessions, and tomorrow he will discuss more in details about the toxicities and maybe his current research and all. Okay, isn't it, Vasant?
Yes, sir.
Yeah, like you are going to discuss in depth tomorrow, right?
Yeah, about
On
yeah. We can tomorrow. We can like we can discuss the basics about how pollution is related to toxicology,
yeah,
and we can
so
know some things about pollution biology also.
yeah, okay. So tomorrow he will be focusing on the pollution biology part, and you just stay tuned for tomorrow's sessions. And tomorrow, the session will be on the same time, like 7:30 p.m. And we will share the Zoom meeting link, just like today, maybe 1 or 2 hour ahead of the meeting. And for those people who are new to the webinar, and this webinar series is a part of our internship program, and all our interns are attending this for the all our interns are sorry okay yeah, all our interns are attending this event for free. Then maybe you can check out our website, and if you are interested in, then you can join our internship program as well. I will share the link with you, and we are delighted to inform you that our internship program is now endorsed by United Nations Decade of Sustainable Development and Ocean Sciences. Okay, so I will show you that. Mm, 1 second. The link is in the chat box. So this is the current course. So this is for the non-interns people. You can check out our website. So if you are interested in, please reach out to us. Now the November intake is quite open. Okay. Okay, then thank you so much. See you tomorrow.